Hello, everyone, and welcome to day six in our African Bioinformatics School, week one. And today we'll be quickly going through the NGS technology overview. The NGS is called the New Next Generation Sequencing, and this is a technology, the latest technology that is being used to sequence DNA or RNA or protein or what have you. So, however, there are some latest technology that we all should know about, and the most prominent among them is Illumina sequencing. Although there are other sequencing that are available, however, one of the sequencing technologies is the Sanger sequencing. So, but it's not the next generation sequencing that is available. That is denoted by that, but it's also one of the most popular sequencing platform that is being used. So, however, Illumina is one of the most popular, and because it is a short read method. So there are different types of method: short read and long read sequencing platforms. So the short read sequencing platforms is Illumina, while the long read is the Nanopore and PacBio. So let's quickly take a look at how, uh, just a brief overview, because you there's no way you would want to work with all the sequencing that you will not have one or two things to. You always be referring back to that terminology. So, how does Illumina sequencing? How does it work? It is sequencing by synthesis light signals from labeled nucleotides. So it's very simple. How Illumina works is that it takes a sequence, it breaks it into tiny pieces. That is the DNA sequence. Breaks it into tiny pieces, and then each of those pieces, they will do a multiple copies of each so that the signal will be strong, and then they will attach each color codes to each nucleotide bases. Either is it A or T or G or C. So let's say A is red, T is blue, G is green, and C is yellow. So the computer will now do that multiple times and take pictures of that color. That color will now determine the arrangement of that color, will determine the arrangement of the sequencing. So that would be like, okay, it's the computer saw the A followed by T, and so that's how the arrangement of the sequencing will be done for all that would determine the order. So there are different processes: library preparation, cluster generation, sequencing by synthesis, and then base calling. So that is basically what I've explained so far with regards how Illumina sequencing works. So the fragments in which they are broken into pieces too is about 300 to 500 base pairs. So that's the breaking into pieces. So attach short DNA adapters to both ends and then allow fragments to bind to the flow cell surface. Now, give that's a rough breakdown, so we can read more on how it works and the technicalities of how it operates. Now, um, there are some I've explained earlier on the short reads and long reads. So, of course, Illumina is the most popular short read and most prominent, and it has its own uh, uses and usefulness. So that's why you have to know which platform you want to use regarding the kind of biology question that you're about to answer. So you have to choose your platform. For example, Illumina, the read length is between 150 to 300. Oxford Nanopore 1000 to 2 million megabits pairs. So read length of PacBio is kind of much more longer. However, their accuracy also varies. The accuracy of all this also varies, and then the cost also varies. Of course, the most expensive is the PacBio, the most expensive. However, what Illumina is best for is for variant calling, for antimicrobial resistant typing, for gene expression, and for metagenomics. And for Oxford Nanopore is used for structural variants, full transcript outbreak surveillance, and This Oxford Nanopore is kind of very little. It's portable and it is usually used. In fact, a lot of lab would have this one in Nigeria itself. I know a lot of labs that have all this, the Oxford Nanopore, because it's portable, very miniature. You can use it on your, like, hold it and then you sequence. So you get your sequencing. So PacBio is much more expensive. It's for reference genome assembly, complex regions, long variants, and the likes. Also, we have what we call the Phred scores. The Phred scores is to determine the quality scores of your sample, of your sequencing. So if your Phred score is 10 or 20 or 30 or 40, you would know that this is what this means. This is what this means. So either poor quality, good quality, excellent quality, you know your Phred score. And it will determine the error inside your sequencing. So you should know what you are working with. So I'm going to send a little bit of quiz and also uh, to share with us what we've learned so far in the first week. There's going to be 25 MCQ questions. You can take 45 minutes and one attempt. Although I've made it easy in such a way that every answer is added at the end of each question, so that we can quickly understand or get the answer that we need. So, but next week on is going to be tougher than this in terms of the answers. So, we are going to grade the answers. So, for week one, we've been able to do for day one, Galaxy Africa, day two, Linux, day three, Python, day four, databases, day five, Blast, day six, NGS. So that is what the week one is all about. So we are looking forward to week two, where we'll be talking about FastQC, BWA, GATK4, Spades, Math, IQ3, mini project, kickoff. So you'll be assigned to a group and then you would get to work on the different projects. So I am glad that we've been able to achieve a successful 1 week, and this is a good start for every one of us. Thank you so much, and see you in the question and answer segment. Bye bye.